Good afternoon and welcome to our 10th INFOGEST webinar on food digestion, live from the Teagasc Food Research Centre here in Moorepark in Fermoy, County Cork in Ireland. This is a special webinar because it's a combined webinar between two networks. That's the INFOGEST network on food digestion and the UNGAP network, and we talk about that later. We are live on the Zoom webinar and on YouTube Live. I'm joined by two excellent speakers, Bart Hens and Michael Grimm. If you can show yourself, please. I'm also joined by Didier Dupont, the chair of the INFOGEST network, and Mirko Koziolek, the chair of the UNGAP network. And I'm also joined by Mern Egan, who is running the whole show from the background. So she makes sure that the whole webinar is functioning well. So without any further delay, I will hand over to Didier Dupont, the chair of the INFOGEST network.
All right. Thank you very much, André. Good afternoon, everybody. Before we start with the presentation, I would like to say a few words on the INFOGEST network, which is the network on food digestion. So as you can see, well, the main objective of the network is to gather different disciplines in order to better understand the mechanism of food digestion. So what we want to do is to develop new models for investigating digestion, including some for specific populations. We want to harmonize the methodologies and propose guidelines for performing new experiments. We would like to validate in vitro models towards in vivo data. We also are very interested in better understanding how bioactive compounds and nutrients are released in the gut during digestion. And we would like to have a better knowledge of the effect of the food matrix structure on the bioavailability of food nutrients and bioactive molecules. So this is how the network looks like at the moment. So we are nearly 600 scientists from 160 institutes in 46 countries. So all the continents are now present within INFOGEST. We have a strong industry involvement. Several companies are following the activities that we have in the network. This is how we are organized at the moment with 6 different working groups. You can see here the coordinators of each working group. So if you want to join one of these working groups, it's completely free and open. So just send me an email and I will be more than happy to involve you in one of these working groups. We also would like, we are in the process of starting new working groups between INFOGEST and UNGAP. So we have identified 5 topics of interest for both network. You can see them listed here. So we would like to work on drug encapsulation with food structures. We would like to work on the application of in vitro models in pharmaceutical sciences. We would like to work together on advanced in silico models. We would like to work on in vitro gut barrier models in order to study permeation, absorption of nutrients and drugs. And we also would like to have some joint activities on drug effects on GI physiology and the effect on food digestion and drug absorption. So if you want to join, feel free to send Mirko an email or to myself as well. Finally, I would like to mention that within INFOGEST, we are organizing a conference, an international conference on food digestion every 2 years. And the next one will take place in Cork, Ireland, between the 3rd and the 5th of May, 2022. So I hope to see you there and I wish you an excellent webinar. Thank you. Mirko,
okay,
would
thank
you like
you,
to say
Didier.
some words?
Yeah, sure. Before introducing the speakers, I would briefly introduce the UNGAP network. So this is a European network that aims to better understand gastrointestinal absorption-related processes. This network has started in 2017 and is a network of scientists from multiple areas, and they all aim to advance the field of intestinal drug absorption. And by doing so, we try to focus on 4 major challenges. So the first is the differences between specific patient populations, such as children or elderly patients. We would like to understand regional differences along the GI tract. We would like to understand the intraluminal behavior of advanced formulation that we see in an increasing number nowadays. And we would like to understand the food-drug interface. So we ask the question, so how does the intake and the digestion of food in the GI tract affect the drug release and also the absorption? And in our working group, we are also scientists from pharmaceutical sciences, medicine, food sciences, and pharmacology, and many more. And we would really like to understand the food-drug interface. And this cost action also aims to advance the career of young and talented researchers from across Europe to really strengthen Europe's leading position in pharmaceutical sciences. And this already brings me to our first speaker, who is Michael Grimm from the University of Greifswald in Germany. Michael currently works at the Center of Drug Absorption and Transport in Greifswald, and he is a pharmacist by training. He obtained his PhD in 2020 from the University of Greifswald under the supervision of Werner Weitschis. Michael is very experienced in applying different in vivo techniques to better understand GI physiology and especially the behavior of dosage forms in the human GI tract. This has led to more than 25 peer-reviewed articles, and in today's presentation, Michael will speak about the gastric emptying under fasted and fed state conditions and its effect on drug absorption. With that, I hand over to Michael for his presentation.
So I just want to say one thing. If you have questions, you can type your questions into the question and answer box at the bottom of your screen. If a few people are on YouTube Live, you have to actually log into your Google account, and you can actually type your question, and we'll answer all questions live afterwards.
Dear colleagues, a very warm welcome also from my side. It's a pleasure to have this opportunity, and in the broad field of food effects, I would like to talk about gastric emptying as a topic of my interest, and I hope it's going to be also interesting for you. As kindly introduced, I'm from the University of Greifswald. Greifswald is a small town in northern Germany, directly at the Baltic Sea, and here you can see our institute, the Center of Drug Absorption and Transport. It's an interdisciplinary building of pharmacology, biopharmacy, and pharmaceutical technology. In the so-called CDAT, we are mainly focusing on oral drugs, and one of our main topics is the influence of food on those oral drugs and how this food can affect the oral drugs' pharmacokinetics. There are many ways of possible pharmacokinetic food-drug interactions. These can be specific, like with grapefruit juice or other specific components of foods, or unspecific. And I want to talk mainly about the luminal conditions, which are fluid volumes and pH and physical chemistry of our gastrointestinal fluids. The luminal conditions can be highly changed by physiological response to the intake of food, for example, increased bile flow or increased secretion in stomach, and all the other changes that are occurring as physiological response to calories. I want to talk about a very specific change, and that is the delay of gastric emptying under the influx of calories and how this can be bypassed. If you are looking for further information about mechanisms of food effects, I'd like to recommend a cited review. There are various literature data available showing that water gets rapidly emptied from stomach, typically between 15 to 45 minutes. We also know that gastric emptying of food is calorie dependent, and typically 2 to 4 kilocalories per minute are emptied. The volume emptying depends on the caloric density, and if we talk about calories and caloric density, when we talk about the fat state, according to clinical trials, we're talking about this 1000 kilocalorie high fat meal that we see here with bacon and eggs and hash browns and milk. And we have seen that this meal is also emptied in a calorie dependent manner, so nearly 3 kilocalories per minute. And that leads to a very, very long fat state, meaning that for approximately 6 hours or even more, we have this fat state persisting. So we would typically expect a very fast absorption in case of fasted intake and a very slow absorption in case of fat intake, just due to the reason of the delayed gastric emptying, irrespective of changed AUCs and other food effects that would people typically would expect. There are several reasons why those typical profiles do not appear, and one of them is the Magenstraße or stomach road. What is this Magenstraße? If we look into an anatomy book, the Magenstraße describes those folds along the lesser curvature of the stomach. And even 100 years ago, people knew that those folds might act as some kind of bypass to form a kind of channel so that water or other liquids can flow around the chyme that is in the fundus and corpus area and directly go into the small intestine. For us as biopharmacists, the Magenstraße is far more than only this anatomical feature. It is a whole phenomenon. And there are computer simulations about motility-driven currents and flows through the stomach by Pal et al., for example. And we did some MRI investigations years ago. And what we have seen there is that the water does not take a distinct channel. It more flows around the chyme and gets emptied from the stomach. So what we see here is a strongly T2 weighted sequence, and the water, the bright area, is emptying the stomach, and even 4 minutes after intake of the water, most of the bright signal is already gone. That was the first description of the stomach road, and I want to go deeper into that now. When I'm talking about first description, I mean direct description by MRI. And that's what we did again in another study that's already published. And we investigated the question, under which conditions and when does this Magenstraße appear? Because the influence might be very pronounced for oral biopharmaceutics. We investigated the effect of timing of water intake as well as the effect of different meals on the occurrence of the stomach road in the study I'd like to present you now. To investigate these influencing factors, we performed a 4-way crossover study with MRI and 6 healthy young volunteers, and our study was closely conducted to the clinical guidelines for bioequivalence and bioavailability testing. We did a 4-way crossover study because we want to compare the fasted state, so the intake of water after an overnight fasting period, with a portion of the standard meal, so high fat, high calorie, and solid components, together with a homogeneous meal, also high fat but no solid components, and a low fat meal, so solid components but nearly no fat. And this way we were able to compare if it's the fat content that plays a role for the occurrence of the stomach road or if it's more the consistency. So we have 2 solid meals with and without fat, and a solid meal and a homogeneous meal, both with high fat content, and all of them were compared to the fasted state. This was our timeline. So after 10 hours of overnight fasting, our subjects took 240 ml of water at time point 0 and also at 60 minutes. This way we were also able to compare if these 60 minutes of ongoing digestion change the occurrence of the stomach road and if the stomach road is also able to appear at later time points. And here I'd like to show you the difference between the high-fat solid meal and the high-fat homogeneous meal. And what we see at the beginning in both cases, we do not have very much of bright fluid in this T2 weighted sequences, only at the border where the secretion occurs in the stomach. And directly after the subject drank the water, we see this bright fluid appearing in stomach. And in case of the solid meal, it nicely flows around the chyme, and we see some sticky boluses inside, which is typical for toast-based meal, for example, and it also flows on top of the meal. In case of the homogeneous meal, we see that the viscosity is lower and the water slightly intermixes with the meal. It does not really mix with it because of the high fat content, but it also entrains the chocolate cream and partially gets trapped under the chocolate cream. So it does not flow on top of it. It more goes inside of it due to the lower viscosity. And 14 minutes later, we see that in case of the solid meal, nearly all of the bright fluid is gone. And in case of the homogeneous meal, lots of the bright fluid is still trapped inside of the stomach, so it has not taken the stomach route, it's still inside the stomach. From the MRI data, we have also calculated the gastric content volumes, and what we see here are the gastric volume curves. For the fasted state, as expected, we see with the intake of water a high increase, that's the ingested fluid, and then a rapid decrease representing gastric emptying. Same happened after 60 minutes again. If we look now onto the 3 different fat states, we see similar curves like compared to the fasted state, representing that also the ingested water taken after a meal gets emptied rapidly, just like in fasted state. To evaluate the differences, we normalize the volumes to the starting volume, and now I'd like to show you these normalized gastric content volumes. So starting volume is always 0, and what we see here are only the water volumes. Now we can compare these 4 curves and having a look on the difference in volume after 30 minutes. So how much volume is still inside the stomach of our ingested water. And in case of the fasted state, it's more or less 0. In case of the high fat solid meal, it's also more or less 0. So nearly all of the 240 ml of water are emptied. In case of the low fat solid meal, the curve is even lower. So there was more emptying than ingested. But in case of the high fat homogeneous meal, we have still entrapped around 70 to 80 and up to 120 milliliters of water inside the stomach. So this is the only meal where we do not have a quantitative stomach road occurring. And now I'd like to highlight the second peaks, because we already knew that for solid meals the stomach road can occur, but what is at later time points? And what we see now is that at later time points there's nearly no difference. So the volume curves are more or less the same, and there are no statistical differences between the first and the second intake of water according to the volume difference, except of one case, and that's the high fat solid meal. Here the gastric emptying of water was even faster at the second intake. So what have we learned from these evaluations? We see that even at later stages of digestion and with smaller portions of meals, the Magenstraße can occur. We've seen that the fat content does only play a minor role for the occurrence of the stomach road or Magenstraße. But on the other side, the consistency and the viscosity and the amount of solid food components is necessary to facilitate the bypassing of water around that chyme. So for us, when we conduct clinical trials, we need to be aware that the Magenstraße can probably occur after all solid meals and for a long time of digestion. But the real interesting point for us is, what does that mean for oral biopharmaceutics? The theory is that the Magenstraße entrains drug substances and can flush it out of the stomach, but is that really true? Will it flush out drug substance? And that's what we have investigated in another study published by Sager et al. And we have compared the gastric emptying, again, of water in fasted state and fat state, and the volume curves are as expected, again with this high fat meal, the FDA standard breakfast. But we have labeled the water with the pharmacokinetic marker, in this case caffeine, and then we have investigated the caffeine pharmacokinetics and measured it in saliva. And what we have seen here, that there are nearly no differences in pharmacokinetics of caffeine in fasted and fat state. If the caffeine would have been intermixed with the chyme, the caffeine curve would have been much, much slower. In this case, we have the pharmacokinetic proof that when we take a solution of a drug substance, this solution just behaves like in fasted state. And if there are no other factors occurring, then we have the same pharmacokinetics. So we have seen that for solutions, the Magenstraße does play an important role, and that the same pharmacokinetic profiles like in fasted state can appear. But what's maybe even more interesting is the influence of the Magenstraße on solid oral dosage forms. And that's what we have done in this study I'd like to present you now. It was a crossover study where we again compared fasted and fat state, in this case with hard capsules. This crossover study was performed in 16 healthy volunteers. The important parts are, again, we have this 240 ml of water at time point 0. And according to EMA guideline, after 120 minutes, we administered 240 ml of water again. And it was a fasted intake and high caloric standard meal. The dosage form was a hard gelatin capsule with 50 milligrams of caffeine and 5 milligrams of black iron oxide to visualize it in MRI. And this is how it looked like. This way we were able to evaluate the disintegration of the dosage form, and also we evaluated the gastric content volume, both in parallel. The pharmacokinetics were again acquired from salivary probes. And this is how it looked like. Again, we have our gastric content volumes, and what we see, we have this typical stomach road profiles. So the water emptying in fat state is just as in fasted state. But this time, the pharmacokinetic profiles look completely different. We have this typical fasted state, rapid increase, but in fat state it was much slower. And what was the reason? Why there was no flushing out of drug substance, just like for a drug solution? Well, for a solid oral dosage form, we have the disintegration as some kind of lag time. And the disintegration was much faster in fasted state than compared to fat state. So the fat state disintegration of the hard capsules was delayed. And this partially explains the delay in onset of pharmacokinetic action. So when we look on our volume profiles, we see that due to the faster disintegration in fasted state, there's still water available that can flush out drug substance, whereas in fat state, most of the water is already gone when the capsule is disintegrated on average. And that partially leads to this flatter profile. When we look into more detail, we see that here we have a second peak in case of fasted state and a first peak in case of the fat state, and that appears directly at 120 minutes. And this is probably mostly related to a second occurrence of the stomach road. We have administered water at 120 minutes, and in these graphs, I'd like to show you how it looks like when you do not administer water at 120 minutes. So on the upper curve, we had a higher dose, and therefore the concentrations are higher. But what we clearly see is that even in fasted administration, we have a second peak when we let our subjects drink water at later time points. And this peak is not there when the subjects do not drink any water again. And in fat state, this second appearance of stomach road also is leading to a flushing out of drug substance and probably entrains the caffeine, which is not the case when you do not administer water, for example. So when we look into the extreme cases of disintegration of the capsules, we see that the longest disintegration took about probably 15 minutes in fasted state, and in fat state even more. That's what I showed you here, and that partially explains what we see here. There's still many drug substance in the stomach at later time points, even in fasted state, and therefore a second appearance of stomach road will face drug substance still there and can entrain it and bring it to absorption. When we look into even more detail into the pharmacokinetic profiles, we see a high variability, and there are specific profiles that are identifiable in this range of variable profiles. For example, this one, which I'd like to show you right now. What's the reason for this profile? We have a very rapid disintegration on top of the chyme. The capsule has contact to the ingested water, and this rapid disintegration in combination with contact to water led to a flushing out of drug substance in very, very early stage, and this leading to a pharmacokinetic profile which just looks like it would have been in fasted state. This is what we typically classify as type 1 pharmacokinetic due to Magenstrasse appearance. There's a type 2 pharmacokinetic, of course, and this is what we see here in this case, exemplary profile right here. And this is related to not that rapid disintegration, so the disintegration is delayed. And what we clearly see, we have no contact to bright fluid inside of the stomach. So there is no pronounced flushing out of drug substance with the co-administered water, but water that is administered at later time points, that does lead to an entrainment of drug substance. Or there's another explanation for those peaks. When a capsule or other solid oral dosage form disintegrates inside of the chyme, there's kind of cloud of drug substance of very highly concentrated API and also excipients. And when this cloud is emptied through the pylorus, we see the effect of what we call gastric dumping. So there is a portion of chyme that is enriched in drug substance, and due to the high viscosity, it cannot diffuse freely inside of the stomach. And this can also cause those peaks. They are not necessarily related to the stomach road. And if we do not have the stomach road and if we do not have gastric dumping, we see profiles like this, what we typically classify as type 3. So when you have a very slow disintegration, no contact to free fluid, and a very, very long residence time of the drug substance inside of the stomach, and it slowly intermixes with the chyme, then we have these flat profiles, which I've shown you in the introduction, what people often assume of typical fat state profiles. As a conclusion, I'd like to show you these 3 types again. We have identified those types of profiles in various fat state studies, and therefore we already came up with a theory years ago. So in short, again, for the type 1, we have the co-administered water that entrains the drug substance in case of rapid disintegration, and therefore we see a rapid onset of drug plasma concentrations. When we have a slower disintegration, but we administer water at a later time point, we can see an entrainment of drug substance at later time points. This can also be related to an effect of gastric dumping. And then we see a rapid onset of drug plasma concentration after a lag time, or when the drug substance intermixes with the chyme, we have this typical flat profile related to the slow and calorie-dependent gastric emptying of the chyme. And what do we learn from this? So with respect to the Magenstrasse, this phenomenon is probably an explanation for early and late peaks in pharmacokinetic profile, and therefore an explanation for pharmacokinetic variability in fat state. The occurrence of the Magenstrasse is possible at all time points in postprandial subjects in case of solid meals, and therefore the administration of water is a very critical parameter for the conduction of clinical trials. So a strict adherence to the EMA guideline might be very critical. For me, it's really questionable because their water ad libitum is allowed at 120 minutes. Therefore, I would really recommend to standardize or to document the water intake in fasted state as well as in fat state. And the best would be if the bioequivalence and bioavailability guidelines would be updated according to this water administration, because it can have this high impact on pharmacokinetic profiles. The dosage form tracking led to even deeper insights. So we have seen that the effects of the Magenstrasse on the pharmacokinetic are strongly dependent on intragastric localization and on disintegration of the dosage form. And this intragastric localization is an additional driver for variability. So this also needs maybe more evaluation. And what we have also seen is that this gastric dumping is an alternative reason for the occurrence of late peaks in a pharmacokinetic profile. So indeed, it's not all related to the Magenstrasse. Of course, I have not performed all this work on my own, and I'd like to thank the enlisted persons right here. Very, very interdisciplinary work of medicine, pharmacology, and biopharmacy. So thank you, dear colleagues. And now I'd like to thank you for your attention, and I will hand over to the moderators for the questions and answers session. Thank you very much.
Excellent talk, Michael. We change over to the live session, live question and answer session. So we have one question that you just answered already. I might read it out again anyway, as it is from Ngandwe Kalungwana. Assuming the rate of gastric emptying is calorie dependent, why would a low-fat diet empty faster than a water-based diet? If you could just expand on that a little bit, Michael.
Yes. Is the sound okay? Yeah. Okay. Great. Well, that depends on what's meant with the question, because we need to be aware that we do not intermix specific questions with each other. So if we talk about gastric emptying of chyme, that's calorie dependent. If we talk about gastric emptying of water, so when we talk about water, then it's mainly dependent on the gastroduodenal pressure gradient. So it's depending on posture and also tension of gastric walls. That's the reason why it's first kinetic. And therefore, if we only talk about water, it's always faster than chyme. But if we talk about water under caloric conditions, so that's what the story of the Magenstrasse is all about, then other factors come into account. So if the volume of the chyme is more, so if the stomach is more filled, we already have maybe a slightly higher tension of the stomach walls. They do relax actively, but when we administer additional water, you need additional relaxation of the stomach walls to form this room for the water to flow around the chyme. And therefore, the occurrence of the stomach road and therefore the speed of the stomach road to occur and the water emptying might be also dependent on volume of chyme that's already inside of the stomach. That might be one reason why we have seen a faster water emptying in case of the low-fat diet, because the low-fat diet with the same calories has more volume, and therefore our starting volume is higher. That might be one reason. And the other reason is we have only quantified volume differences. And with the volume difference is not only water that is emptied. And therefore, during our observation time, we also see caloric emptying of chyme, of course. It's not only water during that time that is emptied, it's also partially chyme. And because the caloric density of the low-fat meal is lower, the calories that are emptied during that time have a higher volume. And that leads that we also lose a bit more volume of chyme during the occurrence of the stomach road. And I think that's what this question was all about.
And I saw there was a glass of milk with a standardized meal.
Yeah, that's true.
Correct, yeah.
Most of our subjects drank the milk in small sips during the meal and not in one flush afterwards, because it's so fatty, it's salty. You need some kind of fluid during eating, yeah, and therefore the milk is probably well intermixed with the meal.
I think that's a general remark by food scientists that find the standardized American meal stomach turning, but it seems to be still the standard. Am I correct in that?
As far as I know, maybe our colleagues from the industry might or can tell us more. But as far as I know, it's still the standard, and it used to be a worst case. Yeah, so there are maybe more representative meals, but it needs to be a worst case. So if we see a food effect with this meal, then we have to evaluate if there are other food effects with other meals. But if we see no food effect with this meal, probably there are no food effects with any other meal too.
Very good. We have maybe 1 minute for a short question, Didier.
Yes, there is one from Ronald Schildering. Do you see any difference in stomach road emptying from a L-shape and a J-shape stomach, as discussed by the University of Leuven? Can you comment on that, Michael?
Oh, that's a very good question. We have not evaluated this different stomach shapes, unfortunately. And as you have seen, we only evaluated 6 subjects, and they were mainly young male subjects. So we had no strong differences in morphology of the stomach. So we were not able to evaluate that. But it's still maybe a topic for the future. That's really a good issue. Yeah.
Good. Thank you.
Okay, so back to Mirko.
Yeah. Thanks, Michael, for the nice talk. I'm now happy to introduce the second speaker, which is Bart Hens from Pfizer Drug Product Design in the UK. Bart is also a pharmacist by training, and he obtained his PhD in 2016 from KU Leuven. He was supervised by Patrick Augustines, who was also leading the UNGAP initiative. After his PhD, Bart was a postdoc at the University of Michigan under the supervision of Gordon Emmerdon, whom some of you may know for his work on the BCS, but also for other topics. He then returned to Leuven in 2018 and then joined Pfizer in November 2020. Bart is very experienced in characterizing the behavior of oral drug products in the human GI tract by in vitro, in silico, and in vivo method. He was very productive with more than 40 peer-reviewed articles, and today he will talk about the luminal behavior of ibuprofen in the human GI tract under fasted and fat state conditions. So Bart, the stage is yours.
Thank you, Mirko. Just wondering how to share the screen because that button seems to be gone. Let me.
bottom of the screen.
Yeah,
Share
to share.
screen.
Oh, yep, indeed, I found it. Thank you so much. Sorry. Perfect. I'm going to the presenter mode. So welcome everybody. And first of all, I would like to thank the INFOGEST organizing team for giving me the opportunity to present today some data related to the, as you can see in the title, the luminal behavior of ibuprofen in the human gastrointestinal tract under fasted and, of course, because of interest at INFOGEST, under fed state conditions. Everybody can see my screen. Can somebody say maybe yes?
Yes.
Okay, perfect. Thank you. First, a little bit of intro, as mentioned by Mirko. I'm a pharmacist by training, did my PhD at KU Leuven. Afterwards, went to the University of Michigan for a postdoc. And at the moment, I'm working at Pfizer UK in the preclinical stage of drug development. So what I'm doing on a daily job is actually, if we have different projects, we have so many compounds in our pipeline, then I am responsible for looking at the molecular properties of those molecules. And the question that they will ask me is what kind of formulation, Bart, will be used if we want to go to the clinical stage to test a drug product in patients or in healthy subjects. And what can we expect from this formulation after oral ingestion? What kind of systemic exposure will be reached? So what kind of concentrations in blood after oral intake? Those are the questions that I need to handle on a daily basis. So how can I actually inform people from the clinical stage which formulation they should use? Therefore, I'm actually relying on in vitro dissolution tools, as you can see. And what we do with those in vitro dissolutions tools is we will actually bring the oral formulation, the candidate formulation, into a dissolution setup, which is in this case like a one-stage dissolution vessel. And actually this pedal, as you can see, will start to rotate and the drug will be released and will start to dissolve. By taking samples from the dissolution vessel, actually, I can measure the concentration that will be released as a function of time, as you can see in this figure. After that, you will see that this dissolution profile is actually giving me an idea of the dissolution rate of the drug, how fast the drug is dissolving in this dissolution media, which is mostly mimicking the media as present in the intestinal tract at pH 6.8, for instance. That's the neutral, almost neutral pH that we know present in the intestinal tract. So I have an idea about how fast the drug dissolves, and I bring that dissolution rate into a computer software program, which is a physiologically based pharmacokinetic software program, for instance, SimSip or GastroPlus. And what this program will do is it reflects the human physiology from a person, could be a healthy subject or a patient. And by putting in the dissolution profile, and the program is already loaded with that physiology of the human subject, I can simulate how much of the drug will appear in the systemic exposure of that person. And then I can inform people from the clinical stage by saying the drug will have this kind of bioavailability. It's definitely a therapeutic effect. It's not toxic. That is actually how I need to inform the people from the clinical stage. The question that you could ask is, what kind of physiological parameters are having an impact, for instance, in the human GI tract on the drug release? And to find out, actually, during my postdoc at the University of Michigan, which was funded by the US Food and Drug Administration, we did an intubation study. And that intubation study was set up in order to examine the behavior of ibuprofen in the human gastrointestinal tract of healthy subjects. And we did that in fasted state conditions by giving ibuprofen with a glass of water, but also in fed state conditions by giving a liquid meal. We didn't use a solid meal as Michael presented in his previous presentation, because when we will aspirate fluids from the GI tract, the solid meal could actually clog the catheter, and therefore we used a liquid meal. We respected the calories as mentioned also by Michael, and also as prescribed by the US Food and Drug Administration. So therefore we used a liquid meal. To give you an idea about an intubation study, because you could ask, some people are maybe not really aware of what's really an intubation study. During my PhD, as you could see, this is me. I was volunteer for these kinds of studies. And what will happen during an intubation study is that you will come to the hospital. Mostly you are fasted, so you did an overnight fast. And the nurse or the MD will actually intubate your nose, or could also be through the mouth, will intubate you with a catheter. And that catheter will be going through your nose or mouth and will be placed into your human gastrointestinal tract. Then you go to the fluoroscopy room where you can actually still push the catheter more inside your human gastrointestinal tract. And as you can see, what you can do is you can position the catheter wherever you want, because of there are some tracers on the catheter that will be reflected on the fluoroscopic image. And so you know perfectly actually where your catheter is situated. And so you can go, even if you want, this is the stomach, and then even you can go further down, you can go to, after passing the pylorus, you are in the duodenum. Then you can go even further, you can pass, as you can see here, this really nice corner is the ligament of Treitz, and then you come into the proximal jejunum. You can go even further then, more distal, and go to the mid-jejunum, or even, if your catheter is long enough, it could go to the ileum, for instance. After positioning the catheter, you go back to the hospital room. You can actually get the drug product of interest. In this case, in Michigan, it was ibuprofen, an immediate release tablet, 800 milligram dose. And at certain time points, as a function of time, you can aspirate fluids from the different places in the human GI tract. So this is actually a small video that shows you the aspiration of fluids. In Michigan, we used one catheter that was able to aspirate at multiple stages, at multiple places. So for instance, in the stomach, duodenum, and so on. But in Leuven, during my PhD, we used multiple catheters to aspirate at different segments. This case, this is human gastric fluid. You can see it. It's it looks very familiar with water. Although if you would measure the pH in fasted state, it's a pH 1 to 2. It's quite acidic. It's also quite viscous, more viscous than water. And this is the aspiration of duodenal fluid. So this is, as you can see, yellow. They're not that viscous as the gastric fluids. And the pH is around 6 to 7. Could be even sometimes lower because of incoming acids. I said that the duodenal fluids are actually yellow. This is because of the bilirubin that is secreted from the gallbladder into the intestine. That's a kind of yellow color given to your intestinal fluids. Just briefly, this is the catheter that was customized by us. We actually developed this catheter ourselves in collaboration with the company MUI, a Canadian company that helped us with the development of the catheter, where you can see the different colors. Red colors are actually places where we have a motility channel. So there was water flowing at a constant rate through the catheter. And whenever the flow of that water would be interrupted, that was given a signal was given on a computer, and we know, like, here's a contraction ongoing, for instance, in the stomach, or in the duodenum, or in the upper jejunum, or in the later jejunum. So a constant water flow, 1 milliliter per minute, was flowing here, and whenever that signal would be interrupted, we know that because the catheter was connected to a computer also, and so we know, like, here's a contraction ongoing. The yellow holes, as you can see, are air channels, because sometimes it's really hard to aspirate a fluid, but by giving a little bit of air, it's easier to really suck up the intestinal fluid, even from the more distal parts, like in the jejunum. Also, we have the blue channels, which is the aspiration channels, of course. And at the bottom of the catheter, we had a balloon channel. Therefore, so there was a balloon here, because if we inflate that balloon, it easily migrates through the intestinal tract. It's easier for us to position the catheter in the more distal parts. Also, here is a tip weight. It's also easier for us then to migrate the catheter to the more distal parts of the human gastrointestinal tract. As mentioned before, where we're working with the drug product of interest was actually ibuprofen, 800 milligram dose from Dr. Reddy's. This is the structure. It's an acidic compound because of the carboxylic acid group, as you can see over here, which means that it has a pKa of 4.8. If the drug will be present in the stomach, that's the first compartment, because of the acidic pH, the drug will not dissolve. It's in its neutral state, the molecule. But when it goes to the intestine at pH 6 to 7, this proton at the end of the molecule will be gone, and it's actually a negative charge, which will make it easier for ibuprofen to dissolve, giving it more polar characteristics. And so the drug will dissolve better in the intestinal environment at pH 6 to 7. Permeation through the intestinal wall is really high for ibuprofen. And so, as mentioned before, it's a Dr. Reddy's tablet coming from the company Dr. Reddy's. This is just a kind of schematic illustrative overview of the study. The 800 milligram tablet was given after the catheter was positioned in the human gastrointestinal tract. It was with water, that's the fasted state, or as I mentioned before, with a liquid meal, which was pulmocare. And so after administration of the tablet with the water or with the liquid meal for the fat state, we started to aspirate fluids from the different segments. Also, blood samples were taken to actually monitor the systemic exposure of the drug. And as mentioned before, also the catheter was connected to the computer for those motility channels in order to actually explore, monitor the gastric contractions as we have observed. 20 people were included for the fasted state studies, 17 people were included for the fat state studies. Interesting, as mentioned before, we were really interested in those motility aspects. The reason why is because we know actually that in fasted states, there are 3 different phases of motility. The entire motility process is called the migrating motor complex, and you have a phase 1, a phase 2, and you also have a phase 3. The phase 1 is actually the phase where you don't have many contractions going on. After a while, you go to a phase 2, more contractions are coming. They're not really well organized. So it's the same as you are sitting in your office. You haven't been eating for a while and you start hearing rumbling of your stomach. That's like phase 2. And when the rumbling goes really hard and it's quite organized, you can have like every 5 seconds you can hear something going on in your stomach. That's what we call the phase 3. That's the hardest contractions we have. And that's what we also call the housekeeper wave. It's like if you have those kind of contractions in your stomach, it's like everything will be directly emptied, like with a broom, out of your stomach into your small intestine. And so those are the hardest contractions you can have. In the fat state, it's not like you have those contractions, phase 1, phase 2, or phase 3 immediately. But so what will happen is that first you have a fat state, everything is actually digesting in your stomach, and as everything will be emptying at a certain speed, as Michael also showed, after everything almost being emptied, the phase 1, phase 2, phase 3 will come up again, like in your fasted state motility pattern. And these are the results from the study. After assembling all the data, because we aspirated fluids, we measured concentrations of ibuprofen. We took blood samples. We also measured concentrations of ibuprofen in those blood samples. And then we also had the motility data. We found actually a correlation between the plasma Cmax and the time to phase 3 contraction. It's not really, you see there's a lot of variability, of course, but we found actually, interestingly, that if we look on the y-axis, we have the plasma Cmax, so that's the highest concentration we reach of ibuprofen in blood of the subjects. And we look on the x-axis, we look to the time to whenever phase 3 contraction occurs after dosing. We see that actually when people were having a high plasma Cmax concentration, we saw it actually also that they were having a really fast phase 3 contraction after ingestion of the formulation. So the phase 3 contraction was really important to determine the plasma Cmax. You see that the blue dots are actually those are the facet state data. Those are more situated on the left hand side of the graph because, as I mentioned before, the phase 3, the phase 1, phase 2, and phase 3 will come up earlier in a facet state. And for the orange dots, it's more situated on the right-hand side because, of course, as I mentioned before, first digestion is needed, and then you will actually have, again, that phase 1, phase 2, and phase 3 is coming up. Another interesting result we obtained from this study is that when aspirating the fluids, we not only measured the concentration of ibuprofen in those fluids, but what we also did actually was we measured the pH. And as you can see in the fasted state, as expected for the stomach, we have quite low pH values. In the fed state, we actually have rather higher pH values because of the liquid meal has a pH around 6. So in the beginning, it's definitely more higher. It's higher and will then decrease after as a function of time. In the duodenum and the jejunum, as you can see on average between fasted and fed state, it's more neutral, going up even to 5 to 6. And more variable, as we can see here, than for instance in the jejunum, because the acid is coming in from the duodenum, from the stomach into the duodenum, and there is some time needed for the duodenum to calibrate and to buffer actually the incoming gastric fluids, and the buffering effect will be occurring by the pancreas, who is actually secreting bicarbonate, and also mucosal cells that are actually secreting bicarbonate. Interestingly, is that the time when we see the Tmax, so the time when we see the Cmax showing in blood, that's at that time we call the plasma Tmax. We had a nice correlation actually with the jejunal Tmax. So it seems that the jejunum, so a little bit more distal in the intestinal tract, was a really important segment where dissolution took place, where the drug started to dissolve. And whenever we saw the highest concentration in the jejunum, at the time we saw the highest concentration in the jejunum, that nicely correlated with the highest concentration also observed at the time when we saw the highest concentration in blood. So therefore, we saw a really nice correlation between plasma Tmax, as you can see on the y-axis, and the jejunal Tmax on the x-axis. The reason why we saw so much dynamic pH changes, because in our in vitro settings, we mostly work with really static pH. We saw actually that the buffer capacity of the fluids was quite low, and that could be one of the reasons why we see such dynamic variable pH values. We measured buffer capacity of those aspirated fluids and compared it with what's already mentioned in the literature. We had really nice, we were in the same range as was reported in the literature. And we know that, for instance, in the dissolution setting, if you go back to the beginning of the slides, when we do a dissolution experiment, we're working sometimes more with higher buffer capacities to keep a more static pH, but we know actually in the human GI tract, it's more variable because of the lower buffer capacity. If you would like to read more about this work that I just presented today, please feel free to go to this paper that is published in Molecular Pharmaceutics. It was a special issue of the Orbito project that ran a couple of years ago. So please have a look at that. And further, I would like to thank the organizing committee, the audience, and if you have any questions, please feel free to ask. Thank you.
Thank you very much. Thank you very much, Bart, for this very nice and clear presentation. We have a lot of questions, so and limited time, so I will ask you for a very short answer. The first question is from Susan Bellman about the catheter that you showed. What was the diameter of this catheter? Can you tell us about that?
Yes, definitely. It's not on the slide, but the diameter of the catheter was 7 millimeters.
7 millimeters. And she also asked whether it could impact gastrointestinal motility, transit, and delivery of chyme. What is your opinion on that?
That's a very good question, and it's a question we get a lot doing these kinds of studies. The answer to that question is that because in the literature we found also PK data from ibuprofen when there was no catheter inside the human GI tract, just from clinical studies where they tested the same drug product but without a catheter and just took blood samples. Our plasma Cmax was in line with the plasma Cmax of those studies. For us, that's a kind of indirect way to think that the catheter doesn't have an impact. What is important, and that's maybe also important to say to the question of Suzanne, is that before starting such a study, a clinical aspiration study, you have to give some time to the subject or to the patient to to calm down, and because in the beginning when you intubate with a catheter, you will see there's a lot of slime coming through the nose. There's more saliva because you are actually activating the parasympathicus nerve system. And that will give you a lot of secretion. So that's really important to give like 15 up to 30 minutes rest to the subject before starting the study.
Okay. George Van Aken would like also to know if this catheter would be available for studying food digestion. Would it be possible, do you think?
Definitely. So I showed my email. You can always contact me, but you can also contact the MUY company, as I mentioned, from Canada. They're not only working with biopharmaceutical scientists like me, but also with people from the food industry to do some digestion studies. So that's also possible to just aspirate fluids, and then you have the fluid after aspiration, and you can do digestion studies, whatever is in your expertise. Yeah.
And maybe one final question, quick question. Again, from George, can the catheter be used to assess the transit rates or luminal pressures? Is it possible?
Well, transit rates are quite difficult unless you maybe would work with a kind of non-absorbable marker. So we did another study where we worked with phenol red, which is non-absorbable and stays in the human GI tract after ingestion. And so then you can follow up how long the non-absorbable marker phenol red will stay in your human GI tract. But it's not labeled like, this is the beginning or the end. So for that, I would actually recommend, and this is more related to the work also of Michael and Mirko, they work with these wireless capsules, and the subject is wearing a kind of transceiver on the outside. And actually, based on that, you can do an outread after, for instance, a day, and you can actually find out how long the capsule was in each segment based on, for instance, pH profiles. You can also look at the motility during the intestinal tract. I think it's the IntelliCap. That's the name of the, maybe Mirko or Michael can say yes or, yeah.
It's the IntelliCap and also the smart pill.
Yeah.
And you could also do it with MRI.
Perfect.
Excellent. Thank you very much, guys. Very interesting. André, would you like to?
Yeah,
give the
excellent.
conclusion?
So yeah, 7mm tube is not for the faint-hearted. So I'd say it takes longer than 30 minutes to calm down after you get a 7mm tube shoved through your nose, But anyway, it was really nice. Thanks for both speakers, Bart and Michael, and also Mirko for introducing the speakers and the network. I just have three more slides, so we finish right on time. So thanks again. There you go. And that's our next webinar. So the INFOGEST webinars are every first Wednesday at 2 o'clock Irish time. Our next speaker is Professor Claire Mills from Manchester, and she will be talking on food allergies and digestion, and no doubt it will be a great talk, and we have enough time for question and answers. It's always a little bit rushed if you have two speakers, but rest assured, if you're watching this webinar afterwards on YouTube, and if you have a question, either contact the speakers directly, contact us, or type your question in YouTube, and eventually somebody will answer it. Okay. Here we go. I mentioned it earlier on, we broadcast these webinars live on YouTube as we speak, but we also have the webinar afterwards on our YouTube channel. So if you go to YouTube, look for INFOGEST webinar, so you eventually find it, or you can scan this QR code. It's a very handy tool. We have 9th is the 10th webinar, but we also have the Food Digestion Conference, day 1 and day 2, that's 6 hours each, with links to the program, and we also have flash presentations right here in the middle. So it's a good source for education. We have the social media. If you want to join the INFOGEST network, Didier mentioned it earlier, write an email to Nathalie Lemar or Didier Dupont, and it's the website. We also have the LinkedIn group. I'm an administrator, so just go to LinkedIn, look for INFOGEST, and at the moment we have 800 followers, so there's a nice, easy tool of following the news on the subject. It's also an easy tool for you to share your publications. So all you do is you basically put a link to your publication and then write @INFOGEST, not hashtag, but @INFOGEST, and the 800 people that follow the site will get this piece of news. So it's a handy way of following, and we will also put up the information on next webinars and where the YouTube channel is and so forth. So until then, 6th of October,